So today we're going to be talking about method validation and how to create consistent processes and products. And this is not only limited to laboratory operations, but we're going to talk about how validation processes can be incorporated into cultivation, manufacturing, and dispensing operations. So today we are talking, I am from PFC, Patient Focused Certification. The Patient Focused Certification Program is a project that was created by Americans for Safe Access. It began in 2014, and what we do is we offer third-party assessments for companies that want to ensure that they're going to pass their state inspections and want to just make sure that they are operating in a safe manner that is going to provide safe medicine for patients as well as create a safe environment for your employees. We also offer pre-state inspections as well as on-site and online training options at 2 different levels, including staff level and verified professional. We're going to be talking today about validation principles. We'll go over a couple of definitions. We'll talk about some different types of method validation, as well as how to apply these principles outside of the laboratory. Some key definitions to start off with. Batch. A batch is usually defined as a quantity of starting material, packaging material, or product that is going to be processed in a single process or series of processes where you expect the result to be homogeneous. And usually this can be done either in a fixed time interval or with a fixed amount. Say you're harvesting a batch of plant, or you're making a batch of chocolate, or you're also running a batch of samples on an instrument in your laboratory. Calibration is a set of operations that establish under specified conditions the relationship between values indicated by an instrument or a different system for measuring, such as a balance, as well as controlling or relating the values represented in a material. We usually calibrate using standard reference materials, as well as you can also use in-house made materials, although purchased reference materials are preferred. The calibration curve should also have acceptance criteria, and we're going to talk a little bit more about acceptance criteria as we get further into this. Contamination is undesired introduction of impurities of a chemical or microbiological nature or of a foreign matter that can end up in your starting material or can end up being introduced during production, sampling, packaging, storage, or transport. Good agricultural practices, also called GAP, result from voluntary audits that verify that the agricultural products produced were packaged, handled, and stored as safely as possible to minimize the risk of hazards, including contamination. Good manufacturing practices, also called GMPs, are also part of the quality assurance process, which ensures that your products are consistently produced and controlled by quality standards. These quality standards are defined in standard operating procedures as well as in training protocols that are, introduced in the facility to ensure consistent processes. Specification is a list of requirements with which the products or materials used or obtained during manufacture have to conform. These are the basis for your quality evaluation, and this, especially when you're manufacturing products, is going to be what you're determining as whether items pass or fail. A standard operating procedure, also known as an SOP, is a written procedure that gives instructions for how to perform a specific task within your facility. That can be cleaning, maintenance, operation of specific equipment, how to mix solutions together. An SOP really is the basis for your quality system to ensure that all of your employees are doing everything consistently in the same. And validation is the act of proving, in accordance with the principles of GMP, that any procedure, process, equipment, material, activity, or system actually leads to the expected result that you want to have. In method validation, there are 8 key points that we're going to talk about, and these are consistent no matter which validation protocol you're going through. And these are precision, accuracy, linearity, range, limit of detection, also known as LOD, limit of quantification, or LOQ, specificity, ruggedness, and robustness. Accuracy is the degree of agreement of a measured value with the true or expected value of the analyte concentration. This can be whether in a laboratory solution or in a flour, an edible, a tincture, any type of product where you have an expected value or your known potency, cannabinoid content, or terpenoid content. And this is often sometimes confused with precision. Precision is the degree of reproducibility of the analytical method, also the degree of reproducibility of the products that you're making. So accuracy and precision sometimes get intermixed and exchanged, but accuracy relates more closely to, is it close to the value that you expect? And precision is, can you do that over again? Specificity is a degree of the measure of interference or absence thereof in the analysis of complex sample mixtures and is determined by comparing test results from the analysis of samples containing impurities, degradation products, or placebo ingredients with those obtained from the analysis of samples without impurities, degradation products, or placebo ingredients. This basically means, is your method actually detecting what you think you're detecting, or do you have something else that's interfering with it? Linearity is usually expressed in terms of variance around the slope of the regression line that is calculated according to an established mathematical relationship. Linearity you generally want to have be as straight line as possible when you make a calibration curve, but you can also use different regressions to force that line to be straight, and ideally you're using the simplest calculation possible because that means that your linearity is more true. The range of an analytical method needs to be established in order to verify the applicability of the method and the analytical technique at widely divergent levels of analyte concentration. When you're doing development work and developing a method for testing something like THC or CBD, you want to make sure that the quantitative value that you're listing is found within your analytical range. So in between your limit of quantification, you have the range. So you have a lower limit and an upper limit of quantification where those values are going to generate an actual result that can be considered quantitative versus a qualitative result which would fall outside of your linear range. The limit of detection, as we just talked about, we just talked about limit of quantification, but the limit of detection is the lowest level of analyte that you can actually successfully detect. And this is typically determined by evaluating the signal to noise ratio on your instrument. And as long as you can successfully identify that, typically it's a 2-to-1 ratio of the signal to noise ratio, which shouldn't be confused with the limit of quantitation. The limit of quantitation is, as we talked about, falls inside that linear range versus the limit of detection, which really just tells you, yes, I've detected this compound, but no, I cannot quantify it. Robustness looks at a variation in optimized conditions. So when you're in your laboratory and you have your instrument, there are a number of different things that can affect the outcome of the result, including variability in the instrument, variability in the analyst, also variances and influences in different chemicals if you've got a new lot of chemicals or you've ordered from a new supplier. That might cause a difference in the results. And if a robust method is really one that's able to be transferred to different locations and across different instruments. There's a number of different validation types. For us, we're going to talk today about laboratory, manufacturing, cultivation, equipment, and cleaning types of validations and ways that you can incorporate the validation processes into that. There's also a number of different validating guidelines present, including those by the International Council of Harmonization, the Food and Drug Administration, the Environmental Protection Agency, and the United States Pharmacopoeia. There are other validating guidelines out there, and it really should be taken into account what your process is to determine which guidelines you should follow to determine how you best to validate your process. In analytical method validation, you need to answer a number of questions first before you can decide what you want to do. What analytes are you going to be validating? Are you validating cannabinoids, terpenes, residual solvents, pesticides, heavy metals? You can also validate microbial species, but based on the analyte that you're going to be validating, that is going to essentially determine how you choose your analytical method. So we know, for instance, that cannabinoids are best evaluated by HPLC. Terpenes are best evaluated by GCFID or GCMS. Heavy metals are best evaluated by ICP-MS. There are a number of different methods out there. A lot of instrument manufacturers have already developed methods for determining some of these compounds. So it's best to start there and then really kind of work your way out. And then if you require a faster method, developing and changing the different parameters so that you have a faster runtime. Maybe you increase your pH ramp. But after you have developed your method, that is when you're going to start doing the validation protocol. And this will be selected. Sometimes it's based on the state that you live in, may have different requirements for validating a process, and depending on sometimes the compound that you're looking for. For instance, EPA governs pesticides, so it would be best to follow an EPA validation protocol to validate a pesticide method. The International Conference on Harmonization, also collectively known as ICH, has a validation of analytical procedures. And there's some questions that you want to ask yourself as you start to review, such as the specificity. Is the assay specific for the analyte? You can't necessarily pick up terpenes in a cannabinoid method. You can probably see some of them, but not all of them. So you want to make sure that your assay is really specific to the analyte that you're looking for. You also want to have good linearity, so your signal versus your concentration. If you're having to adjust your linearity and change how you're calculating your regression line, it may be that you need to evaluate your method a little more so that you can get a linear line that is linear and not one that has to be really heavily played with to get it to meet the acceptance criteria. Your range is generally derived from your linearity studies and will also help you determine your LOD and your LOQ. Precision and repeatability. For ICH, this is going to require that you do 9 determinations over your concentration range. So for example, you're going to do 3 concentrations, 3 replicates of each of those concentrations, or you can also do 6 determinations at 100% of your test concentration. This really just depends on how much time you have and how deeply you want to go into the validation process. Typically, this type of a validation should really only take about a day to perform as long as your calibration and your instrument are operating optimally. You're also going to want to use fresh calibration standards and fresh diluted standards for whenever you're doing any of these studies. Reproducibility is typically done by interlaboratory studies for standardization of an analytical method. However, if you have multiple instruments in your laboratory, you can also show reproducibility by being able to do the exact same thing on multiple different instruments. This is also going to go alongside the ruggedness and robustness of your method. Can your method be carried over into other laboratories, or can your method be carried over into other instruments? And it's not just specific to the one thing that you're doing. In the ICH, they also define your limit of detection and your limit of quantification. And your limit of detection is really evaluated by either your signal to noise with a background noise of 3 to 1. Also can be calculated by your standard deviation of the response and slope, which is this formula over here. It can also be looked at with the standard deviation of your blank as well as your calibration curve. So all of these things should be factored in when you're determining your limit of detection. And similarly, with your limit of quantification, we want to have a higher signal to noise ratio so that we know that background noise is not what we're actually calculating, but you're actually calculating a true result from the true analyte that you're detecting, as well as taking into account the standard deviation of the response and the slope, the standard deviation of the blank, and the calibration curve. Accuracy, not to be confused with precision, should also be established across the specified range and done with 3 concentrations and 3 replicates. This can be done in a single day, or it can be done over the course of multiple days. You should be using an analyte of known purity, so purchasing a pure reference standard or making one yourself that you have been able to verify is pure. You want to compare your results to those of a well-characterized procedure. So in that essence, when you're buying a standard, it should come with a certificate of analysis. That company that is manufacturing it should have a developed method to determine the concentration of the standard that they're giving you so that you can compare the results from your analysis to that of the certificate of analysis and ensure that your instrument is reading your standards correctly. Once you have inferred your precision, your linearity, and your specificity, you can determine how accurate your method is. Robustness should show the reliability of analysis with respect to deliberate variations in method parameters. Some of your typical variations could include the stability of analytical solutions or how long you're doing a sample extraction for. In other areas, such as liquid chromatography, you can have variations in your pH of your mobile phase, which could cause a change in analysis, as well as column composition. If you've purchased a new column or you're switching between columns, you want to make sure that you're validating for each type of column that you're using, as well as temperature and flow rate. And similarly with gas chromatography, your column composition, your temperature, and your flow rate can all affect the outcome of your analysis. For the FDA, their validation protocol they have a number of them, but their bioanalytical method validation guidance for the industry is one that is generally followed. The FDA builds upon what the ICH does. A number of things are the same, but they also want to ensure that your reference standards are of known purity and concentration. So you're not making them in-house, or you've verified them outside of in-house testing. Critical reagents, making sure that you know the identity and the purity of everything that you're doing. So if you're buying methanol from a vendor, you want to make sure that you're verifying that that lot of methanol is not contaminated and wouldn't cause any effect on your analysis and your results. So you need to evaluate these reagents prior to actually putting them into use in your laboratory. Your calibration curve. Is it continuous, and is it reproducible after validation? So if you've had a successful validation and you've had this great calibration curve, what happens when your calibration curve starts to go out? Can you reproduce this calibration curve? Selectivity and specificity. This is during the validation, the potential for the analyte interference should be determined. So is there background interference? So for example, we know that hops shares a number of similar qualities with cannabis. They're in the same family. However, hops has a lot of background interference that would interfere with a cannabinoid analysis. And so you want to make sure that you're not having interference. And also edibles. Edibles are a big thing. There's a lot of different items in that matrix that you need to evaluate. Is it causing background noise, and can you effectively evaluate the analyte that you're looking for? Accuracy and precision, this is one of the ways that the FDA builds upon ICH. They have a minimum of 3 to 6 replicates at different concentrations. They must be run over several days to show that you can actually reproduce the same results over and over and over again. The stability of the analyte inside a matrix should also be evaluated for standard storage conditions and if storage conditions change. That's already been done for reference standards that you're purchasing. Your certificate of analysis will tell you exactly how you should store and when that expiration is. When you're developing new products like topicals or edibles, And even your flowers, you want to do stability studies to ensure that you are telling people how to properly store your product so that you don't cause degradation or potential contamination. Once you have validated a method, if you change a condition, such as your pH, you may only have to do a partial validation instead of a full validation. So if you changed one reagent, then you would only have to revalidate the parts that include that reagent. The FDA also defines acceptance criteria. So for your calibration curve, you must be plus or minus 15% of the non-zero calibrators and plus or minus 20% of the lower limit of quantification. For your selectivity and specificity, your spiked samples should be plus or minus 20% of the expected value. For accuracy and precision, they must be plus or minus 15% of the calibration values and plus or minus 20% of the calibration value at the lower limit of quantification. The FDA or the EPA has their own validation. It's called Validation and Peer Review of the U.S. Environmental Protection Agency Chemical Methods of Analysis. This is typically for in-house developed methods that are done within the EPA. However, they also can provide some guidelines for when you're working on a method that maybe fall under EPA guidance, such as analysis of pesticides. And some of the differences here or some of the definitions for them is they always want to make sure that you're correctly identifying analytes. And of particular concern, of course, is if you're looking for pesticides, you really want to make sure that you're correctly identifying them, because a false identification could lead to a lot of trouble for people. Instrument calibration, you want to make sure that your response versus your analyte concentration is linear. They also want to make sure that you know the bias and what the magnitude of that bias is, as well as the magnitude of any random errors that might be associated with the method. They also want to ensure that you're determining your limits of quantitation and range, as well as your detection limits. The United States Pharmacopoeia has Chapter 1225, which is the validation of compendial procedures. They also reference Chapter 21 of the Code of Federal Regulations, and they also reference the International Council on Harmonization. So there are a number of different validation protocols out there, and it's really up to you to figure out which protocol should you follow that best meets the needs of what you're trying to do. And some of the ways that we can transfer this into businesses that aren't necessarily laboratories is by transferring some of these principles. So for example, if you're validating your equipment, let's say you've purchased a brand new piece of extraction equipment, you need to validate that you are actually extracting everything out of the flour that you're putting in there so that your oil is consistent every time. So accuracy, is the cannabinoid or terpenoid content outcome as you've expected? So by doing multiple runs, you're going to be able to determine, did you do this? Also, are you trying to decarboxylate inside the piece of equipment? So is that outcome actually happening? And a lot of this is determined by doing testing. And so testing your cannabinoid content or your terpenoid content. You also want to ask yourself some questions regarding precision. Is it consistent from product to product? Can you receive the same results batch to batch? What happens if you change something in that process? What happens if you use methanol instead of ethanol? What happens if you use wet flour versus dry flour? You want to know how those changes might cause your outcome to be different. You also want to determine the stability, how do changes in your environment. For instance, if you work somewhere where there's fluctuations in temperature, does it snow? Does it get really hot in the summer? Is it really humid? How do those changes affect the equipment? And also, what is your acceptance criteria? If the temperature outside is too hot and it affects your equipment inside, what is the permitted deviation that you can have? How do you determine if you need to adjust your equipment or adjust your method? What is acceptable and what is not? And similarly, when you're cleaning your equipment, you also want to be asking yourself the same questions. Is the equipment or the room cleaned in a way that doesn't cause cross-contamination? Is it cleaned the same way every time? Is it cleaned the same way every time by each different person performing that task? What changes in the procedure could cause your outcome to be different? Did you hire someone new? Did they not follow your standard operating procedure? Did they take it upon themselves to do something a little different that maybe not is included in your SOPs? How do your environmental changes affect the results of the cleaning? What is the permitted deviation on this? Is it okay to have a little bit of soil or oil left in your pot or in your extraction equipment? What is allowed? The same thing can be put into cultivation process. Is your cannabinoid or terpenoid content consistent with your historical data? Is it consistent plant to plant? Is it consistent batch to batch? What changes in your cultivation process could cause the outcome to be different? Did you start purchasing nutrients from a new supplier? Did you install a new fertigation system? How do your terpenoid and cannabinoid contents trend over time? What is the permitted deviation? How do you know when a mother plant is time to be retired because your clones are losing potency? What is that limit? And in manufacturing, a similar thing also. Is your products consistent with the historical data? Is the cannabinoid or terpene content consistent from product to product or from batch to batch? What changes in the manufacturing process could cause the outcome to be different? Did someone call off sick and you had to put somebody into a new process that maybe they're not familiar with? How is your training making sure that your employees are performing these tasks consistently in the same way every time? How do your content values change over time? And what is your permitted deviation? And particularly in manufacturing, this deviation is sometimes defined by the state that you're in. Maybe they only allow a plus or minus 10% deviation on your label value. Maybe it's 15%, maybe it's 20%. But what is allowed and how do you ensure that you're within the guidelines of your acceptance criteria? One of the things to keep in mind as you're pulling samples from your cultivation or from your manufacturing facility is really how you're taking your samples. And this can be fairly critical. One thing you want to do is really possibly, if you can, have a statistician come in and really develop a sampling scheme based on the size of your grow or how many products you're manufacturing in a day or a week or a month, so that they can help you determine when and where to pull samples to ensure that everything is coming out and you're being able to identify, did something deviate from my process? Is something going to fall outside of my accepted range? For data analysis, you really want human eyes to be on this. There are many softwares out there that can analyze your data and view your trends and give you ideas, but nothing will beat a trained eye that knows how to look at this data and knows, did something deviate? Is something going to deviate? You want to really ensure that you have all of this determined. Randomness. Ideally, there should be an equal likelihood for any unit being sampled. So did you pull a half-gram sample from 10 pounds of bud? Maybe you want to pull something different because we all know the variation. Representativeness. Everything in your production should be the same. You should be homogenizing everything. So, for example, if 1/3 of a plant in the population is dark green, then you should have 1/3 of your sample that you're checking also be dark green. So in summary, we have 8 key points of method validation. We have accuracy, precision, linearity, range, limit of detection, limit of quantification, specificity, ruggedness, and robustness. These principles are the defining feature various validation protocols, including ICH, FDA, EPA, USP, as well as many others. And you can apply all of these principles and protocols to different areas, and it's not just laboratory. You can apply it to your equipment, you can apply it to cleaning, you can apply it to your cultivation, you can apply it to manufacturing. I'm going to check and see if there's any questions right now, but if anyone has any questions, please feel free to type them into the chat. Otherwise, there is a discount for our online training. At PFC, we offer online training at a staff level and verified professional in cultivation, manufacturing, dispensing, and laboratory operations. We really like to recommend that most employees of the facility get at least the staff level training. And then really, if you want to include the verified professional, that adds on additional courses, not just for managers and owners, but that could also benefit employees. And it doesn't look like we have any questions, so I would like to thank everyone for coming in here today and joining us on this webinar. Please go visit our website. You can also email me directly if you think of any questions that you have at the end or later today, tomorrow. You can reach me at heather@safeaccessnow.org. Thank you very much.